Hi, my name is Amar Sahay, and my laboratory is at the Center for Regenerative Medicine at Massachusetts General Hospital at Harvard Medical School in Boston. As many of you may be aware, the hippocampus plays a critical role in the formation of episodic memories, memories of what, when, and where. And the computations that the hippocampus performs about the world around us, that's our context, is used to then calibrate the activity of subcortical circuits that subserve physiology and adaptive behavioral responses to our changing world, our context. It's intuitive to think how hippocampal dysfunction is a central component of cognitive disorders, such as age-related cognitive decline, Alzheimer's disease, as well as psychiatric disorders that are characterized by hippocampal dysfunction. In my laboratory, we're interested in a unique form of hippocampal circuit plasticity, adult hippocampal neurogenesis, the process by which dentate granule neurons arise from these radial glial neural stem cells in the dentate gyrus of the hippocampus throughout life. In a complementary line of work in my lab, we spend a lot of time thinking about this wiring diagram of the hippocampus. This is a simplified wiring diagram, and the causal role of connectivity motifs, elemental features of this wiring diagram, to the functions of the hippocampus in memory and the regulation of emotion. And our hope is that these efforts may inspire strategies to rejuvenate the dentate gyrus with new neurons in disease states, restore or repair the hippocampus, and re-engineer circuit motifs to optimize hippocampal memory processing. And I'll give you some examples along the way in the next few slides. The neural stem cells in the dentate gyrus, shown over here, once they become activated from a quiescent state, can divide symmetrically to make copies of themselves and expand the stem cell pool, or divide asymmetrically to generate a copy of itself and an astrocyte, a process called astrogenesis, or divide asymmetrically to generate a copy of itself and a progenitor, a neural progenitor, which then becomes or differentiates into an adult-born neuron over a protracted period of time, which then integrates into the hippocampal circuit to influence hippocampal functions. In my lab, one line of research is aimed at understanding the mechanisms that regulate neural stem cell activation and quiescence, a fundamental decision that has a lasting impact on the hippocampus. Recently, we have identified a transcription mechanism that regulates symmetric stem cell divisions. So in other words, a mechanism by which stem cells make more copies of themselves at the expense of astrogenesis or neurogenesis. And you can see here in the top right, with longitudinal in vivo 2-photon imaging, stem cells undergoing asymmetric or symmetric divisions. And here, 2 radial glial neural stem cells. We're interested in how these mechanisms may be harnessed to replenish the neural stem cell pool in aging, as well as in different disease states. But the neural stem cell is very influenced by the cell types that surround it or ensconce it, what's referred to as the niche. The niche of the neural stem cell is made up of microglia, astrocytes, endothelial cells, and the niche is known to change over time in aging, such that it becomes unhealthy with regards to its ability to support the stem cells, as well as synapses in the dentate gyrus. Efforts in my lab are underway to identify signaling mechanisms used by neurons in the dentate gyrus to communicate, much like an orchestral conductor, with these different cell types, microglia and astrocytes, to ensure that the neural stem cells are preserved, as well as synapses, during aging. As I said earlier, We're also very interested in the circuit mechanisms that govern the integration of adult-born neurons into the hippocampal circuit. A few years ago, we identified a role for synaptic competition in governing the extent to which these young adult-born neurons have to compete or successfully compete with pre-existing mature neurons. And by identifying a molecular factor that governs this competition, we were able to toggle the switch and rejuvenate the dentate gyrus of aging animals with extra new neurons that had won this synaptic competition. We're now following up on these studies with signaling pathways that we have identified that we think play an important role in the pruning of input connectivity and output connectivity of dentate granule neurons. And these studies may generally edify how we think about pruning in the adult brain. A major effort in my lab, as I said earlier, is looking at that wiring diagram of the hippocampus and trying to identify a causal role for circuit motifs in the functions of the hippocampus in memory processing. A few years ago, we identified an important role for adult-born neurons in the dentate gyrus in the discrimination of similar contexts by governing population-level activity in the dentate gyrus. We found a role for oxytocin receptors in the dentate gyrus CA3 circuit in governing the discrimination of social stimuli. We identified a role for feed-forward inhibition in the dentate gyrus CA3 circuit as critical for dictating the extent to which memories preserve details with the passage of time, or time-dependent generalization. We identified a neural pathway that links the hippocampus with the many different subcortical circuits like the BNST, the periaqueductal gray, the lateral hypothalamus, the supramammary nuclei, via the dorsolateral septum to calibrate appetitive and defensive behaviors by regulating the activity of these different subcortical circuits. If you'd like to learn more about the lab or meet the fine folks in the lab who've done a lot of the experiments and are working on many of the projects that I've alluded to, you can stop by our lab website, and we'll be happy to meet with you. Thanks for your time.